Parra protocol can be used to determine spatial and temporal dynamic events during mitosis, and can also be used to visualize the effects of mitotic regulators in real time. Using dual color imaging allows a simultaneous analysis of two molecular markers. For example, we can visualize the microtubules of the mitotic spindle and the kinetochore structure of replicated chromosomes.
To induce fluorescent protein expression 4 days after RNA interference treatment, expose inducible metallothionein promoter transfected cells to a final concentration of 500 micromolar copper sulfate for 24 to 36 hours. To prepare the fluorescent protein expressing cells for imaging, transfer the cells to a sterile 15 milliliter tube and, after counting, sediment the cells by centrifugation. Resuspend the pellet in fresh 25 degrees Celsius warmed SIM supplemented with 10% FBS at a 2 × 10⁶ cells per milliliter concentration, and treat the cells with additional 500 micromolar copper sulfate. Then add 200 to 500 microliters of cells to one well of a multi-well live cell chamber and place the chamber on an inverted fluorescent microscope stage. While the cells are settling in the chamber, open the live cell imaging software and click New and Experiment. To insert a time-lapse loop over which the images will be taken, click the time-lapse loop icon. Set the interval to seconds and divide the desired overall experiment length in seconds by the interval to set the number of cycles. Allow the loop to repeat over the desired total time period. To insert an infrared